So hey everyone, today we are going to see about ion exchange chromatography. So what are the contents we are going to see is introduction, type of ion exchange chromatography, type of ion exchanges, working principle, instrumentation, procedures and application. So introduction. Ion exchange chromatography is a technique for separating compounds based on their net charge. Ion exchange chromatography media contains negatively or positively charged functional groups covalently bound to a solid support, yielding either a cation or an anion exchanger respectively. Charged compounds are adsorbed and retained by an ion exchanger having the opposite charge, whereas compounds that are neutral or have the same charge as the media pass through the void volume and are eluted from the column. The binding of the charged compound is reversible and absorbed compounds are commonly eluted with a salt or pH gradient. Ion exchange media are available in various particle sizes, ionic forms, and purity ranges. Ion exchange chromatography is most often performed in the form of column chromatography. So, what are the types of ion exchange chromatography? There are different types of ion exchange chromatography. Some of them are anion exchange ion chromatography, cation exchange ion chromatography, size exclusion ion chromatography, high performance ion chromatography, ion chromatography with suppressed conductivity detection, ion chromatography with amperometric detection, ion chromatography with mass spectrometry, and chiral ion chromatography. So types of ion exchanges. So in this process, we use two types of exchanges. They are cationic and anionic exchanges. So cation exchanges process negatively charged group and these will attract positively charged cations. So these exchanges are often called as acidic ion exchange materials because their negatively charged results form the ionization of acidic group, whereas anionic exchanges have positively charged groups that will attract negatively charged anions. So these are called as basic ion exchange materials. Principle. The form of chromatography relies on the attraction between oppositely charged stationary phase known as anion exchanger and analyte. The ion exchangers basically contain charged groups covalently linked to the surface of an insoluble matrix. The charged groups of the matrix can be either positive or negative charge. When suspended in an aqueous solution, the charged group of the matrix will be surrounded by ions of the opposite charge. In this ion cloud, ions can be reversibly exchanged without changing the nature and properties of the matrix. Form of working principle for ion exchange chromatography, whereas the charged particles are eluted. So instrumentation. Typical ion chromatography instrumentation includes pump, injector, column, suppressor, detector, recorder or data system. This pump. The IC pump is considered to be one of the most important components in the system, which has to provide a continuous constant flow of the elutant through the IC injector column and detector. So the second one is injector. Liquid samples may be injected directly and solid samples need to be dissolved in an appropriate solvent. Injection should provide the possibility of injecting the liquid sample within the range of 0.1 to 100 ml of volume with high reproducibility and under high pressure. Depending on its ultimate use and area of application, the column material may be stainless steel, titanium, glass, or an inert plastic such as PEEK. The column can be vary in diameter from about 2 mm to 5 cm and in length from 5 cm to 50 cm, depending on whether it is to be used for normal analytical purposes, microanalysis, high-speed analysis, or preparative works. God column is placed anterior to the separating column. This serves as a protective factor that prolongs the life and usefulness of the separating column. They are dependable column designed to filter or remove the particles that clog the separating column. Suppressor. The suppressor reduces the background conductivity of the chemicals used to elute the samples from the ion exchange column, which improves the conductivity measurement of the ion being tested. IC suppressors are membrane-based devices which are designed to convert the ionic eluent to water as a means of enhancing the sensitivity. Fifth one is detector. Electrical conductivity detector is most commonly used detector, and a pre-programmed computing integrator may be sufficient for recording. So next procedure. Ion exchange separations are carried out mainly in columns packed with an ion exchanger. These ion exchangers are commercially available. They are made up of styrene and divinyl benzene. Example, DEAE cellulose is an anionic exchanger, whereas CM cellulose is a cationic exchanger. The choice of exchanger depends upon the charges of the particle to be separated. The separate anions anionic exchanger is used. To separate the cations, cationic exchanger is used. Firstly, the column is filled with an ionic exchanger, then the sample is applied, followed by the buffer. The tris buffer, pyridine buffer, acetate buffer, citrate and phosphate buffers are widely used. The particles which have high affinity for ion exchanger will come down the column along with the buffers. In next step, using the corresponding buffer separates the tightly bound particles. Then these particles are analyzed spectroscopically. So this is the procedure for ion exchange chromatography. So what are its applications? An important use of ion exchange chromatography is the routine analysis of amino acid mixtures. The 20 principal amino acids from blood serum or from the hydrolysis of proteins are separated and used in clinical diagnosis. This is the most effective method for water purification and used for softening of hot water. In the analysis of products of hydrolysis of nucleic acids, they gain about the structure of these molecules and how it relates to their biological function as a carriers of hereditary information. Chelating resins are used to collect the trace metals from seawater, to analyze the lunar rocks and rare trace elements on Earth. Advantages. It is one of the most efficient methods for separating the charged particles. It can be used for almost any kind of charged molecule, including the large proteins, small nucleotides, and amino acids. Ion exchange is used for both analytical and preparative purposes in laboratory, the analytical uses being the more common. Inorganic ions also can be separated by ion exchange chromatography. Limitations. Only charged molecules can be separated, and this method requires buffer.